So today's topic right now, I'm going to teach us about antigen. So, you can you tell me basic thing about antigen? What do you know?
Antigen, it's protein.
Okay.
Like can be like foreign body.
Yeah, exactly. It's mainly the foreign body which enters into the our body and it causes an immune reaction. So that's a function of the antigen. So it has got two main properties. One is immunogenicity and second one is immunological reactivity. What do you mean by immunogenicity is ability of an antigen to induce an immune response in the body. That is antibody production. See, not all foreign bodies are called antigens. It should satisfy these two main properties, that is immunogenicity and immunological reactivity. So in immunogenicity, that is when the foreign body enters in your body, it should be able to create an antibody production. That is what immunogenicity is, and it may be a both humoral and or all cell-mediated immunity. Then immunological reactivity is antigenicity, that is ability of the antigen to react with the antibody. So if a foreign body just stimulates the antibody production alone, it is not called antigen. But it should be, this antigen should be able to bind the antibody which has been produced. So like that, the antigen antibody reaction will take place. That antigen antibody reaction in further you'll come to know. So an example for immunological reactivity is the precipitation reaction, then the agglutination reaction is the example for immunological reactivity. So you got right what an antigen's main property is.
Yes, sir.
Oh, okay, fine. So next is called hapten. Hapten is a defective antigen. It's low molecular weight molecules that lack immunogenicity. Got it? So the immunogenicity is able to produce antibodies called immunogenicity. So haptens are defective antigens because it's not able to cause immunogenicity. Then next is but it retains antigenicity or immunological reactivity. That is, it is able to bind to an antibody if an antibody is produced by something else, not by haptens. It will be like some other antigen, but haptens have a property of immunological reactivity. That is, it is able to bind an antibody, but it can't produce its own antibody. So it can become immunogenic on binding larger protein molecules called carrier proteins. That is, haptens can be immunogenic if it's able to bind a protein called as carrier proteins. So whatever I told is like, see, hapten is defective antigen because it is not able to produce its own antibody. That is, it can't induce an immune response, but it has a property of immunological reactivity, that it can bind to an antibody. So this is the property of hapten. But hapten can become immunologically active if it is binding to a carrier protein molecule called carrier. So you got what hapten is, right?
Yes.
Yeah. Okay, fine. They might ask you this hapten. What is an hapten for it? Like you have 2 mark questions, right? So in 2 mark questions, they might ask you what is an hapten. So haptens are classified as, pardon?
We have 4 marks questions, then 1 mark.
Yeah, 1 mark. So in 1 mark question, they might ask you what is an hapten. 1 mark question, they might ask you what is hapten and how are haptens classified. So now I'm coming to classification of hapten. Hapten is classified into it is like simple hapten, complex hapten. It's classified into 2 types: simple hapten and complex hapten. So in simple hapten it has only one epitope. You know what epitope is?
No, sir.
No.
Sir, it's something. It's part of the antibody, right?
No, it's part of an antigen. So see, now we are talking about an antigen. So this epitope is the antigen binding. It's an antibody binding site on an antigen. Got it? See, when an antigen and an antibody binds, the area where an antibody binds to an antigen is called epitope. So the epitope is on the antigen. It's a receptor on an antigen, which causes the binding of an antigen to the antibody. Yeah, that's much better. You won't get confused. So, an epitope is a receptor on the antigen, which helps in the binding of the antibody to the antigen. Yeah. So, if an epitope is not there, then the antibody can't bind, or antigen can't bind the antibody. Got it? Okay. So this hapten is called simple hapten only when there is a single epitope and it doesn't precipitate. Hence, it can't be visualized. In order to visualize an antigen, it should precipitate. But simple haptens don't precipitate, so it can't be visualized. Whereas complex hapten, it has two or more epitope and it precipitates, so it can be visualized. Got it?
Yes.
Yeah. So epitope is the smallest antigen determinant that binds to specific part of an antibody. Similar to an epitope, for an antibody to bind to an antigen, there is a receptor on the antibody also. It is corresponding receptor is called paratope. See, down I've written paratope.
Yes.
So epitope is on antigen. Corresponding receptor on an antibody is called paratope. Okay. Just a minute. If I close this, can you hear me? Yeah, you can hear me, right?
Yes, sir, I can hear you.
Yeah, okay, fine. I just closed that window there. Okay, now next is factors influencing antigenicity. This is very important. It's a formal question for you. The factors which influence antigenicities are following factors. One is the size of an antigen. Larger the size, more is the antigenicity. That is the more potent. See, the size of an antigen is larger, it is more antigenic, it's stronger antigen. The next is chemical nature. Proteins are more antigenic than carbohydrates, and carbohydrates are more antigenic than lipids, and lipids are more antigenic than nucleic acid. They might ask you question in 1 mark, like or a multiple choice question. Which is the most antigenic out of the following? So you have to know this order. So that the proteins are more antigenic than carbohydrates more than lipids, and lipids more than nucleic acid. The next is susceptibility to tissue enzymes. Antigen is degradable by tissue enzymes. It leads to generation of number of epitopes. That is, when an antigen is exposed to tissue enzymes, it undergoes breakdown, and each broken down molecules will have an epitope, and it will increase the when a single antigen is broken down into many antigens, and each antigen having an epitope, this has become more antigenic. So increased antigenicity is there for this fragmented particle of antigen. So next is example is lactase is not degradable, so it does not induce antigenicity, but L group of amino acids are antigenic and is degraded by the tissue enzymes. The next is foreignness. Foreignness is a key factor because in order to produce an antibody, that antigen should be foreign. If the body doesn't recognize it as a foreign, it won't cause an immune response. So more the foreignness of an antigen, more the antigenicity. Next is genetic factor. Different individuals of a given species show different types of immune response towards the same antigen due to genetic differences between the individuals. That is different. See, like me and you will show different response to a particular antigen. It might not be the same. Example, if like you just take, some people have get affected by COVID, some people don't get affected by COVID, because it's the way the body's response is. So it may be due to the genetic difference in the individuals. Like some people are having good immunity, some people are not having good immunity, or some people have a good immune response, that's why they became COVID positive and showing symptoms outside. Some will be dormant within the body. So optimal dose of antigen, this was a good factor because antigen is immunologically active in optimal dose range. There should be a certain number of antigens for the immune response to take place. If there is like 1 or 2, the immune response won't take place, it will enter and just go off. But there should be an optimal dose for the immune response to take place. Too little dose fails to elicit the immune response. Too large dose leads to development of immunological tolerance also. You just imagine like continuously exposed to the same antigen like chicken pox. You're continuously exposed to the same antigen, it will cause immunological tolerance. So you won't show any symptoms later. And example, one more thing is like COVID vaccination. You keep on like first dose, second dose, third dose. That's what herd immunity comes in action. So like you're getting like exposed to the same antigen, and then you get immunological tolerant to it. The next is route of antigen administration. Immune response is better induced by parental administration. Then the other routes of administrations are peroral and the inhalation. Then repeated dose of antigen. To generate adequate immune response, repeated dose of antigen over a period of time is required. It's the same thing. You imagine like hepatitis B vaccination. After that, in order to boost the immune system, or in order to get an immune system like fully developed, again you keep the hepatitis B booster doses you start giving. Have you taken booster dose of hepatitis B? Have you taken vaccination of hepatitis B?
Yes.
Yeah, you're taken. Okay, fine. So you know that first dose. After that, after 3 months, you take the second dose. Yeah, the same thing. Yes. The next is antigen host relationship. So in this week we'll see the how the antigen and our body reaction takes place. Self or auto antigen. In this self or auto antigen, it belongs to the host itself. It is not immunogenic. That's what I said. In order to in self or auto antigen, the self and body itself recognizes it as a self antigen. So there won't be any immune response. It shows immunological tolerance. That is, immune system does not react to its own antigen. So at that time, when an immune response is not elicited, that antigen is called self or auto antigen. Whereas non-self or foreign antigen is where the immunological response takes place. It's very immunogenic. There are three types of non-self or foreign antigen. One is alloantigenic antigen. That is, it is species specific. But humans will have a specific species of antigen. Animals, in that animals also different species will have, the particular group of species will have particular antigen. That is alloantigen. Isoantigen is the present only in subset of species. In a particular species, only subset group will have this particular antigen. Like you see in humans, there are different types of blood group antigens. That is A blood group, B blood group, AB blood group, O blood group. So within the species of the species, subset species will have different group of antigen. Other example is histocompatibility antigen. Histocompatibility antigen is the one which we see during kidney transplant, organ transplants. Do you know about organ transplants?
Grafting.
Yeah, grafting and all. Histocompatibility antigen is. Next group of non-self of foreign antigen is heteroantigen. That is antigen belonging to two different species. That is antigen of plants or animals or microorganisms. Next is heterophyll antigen. This is very important because heterophyll antigen is an example of a heteroantigen. It is coming under heteroantigen, but it is given a separate group because it's bit of importance for us. It's a type of heteroantigen. It is cross-reactive antigen. It's present in two different species, but they share the same epitope with each other. That is, humans and the plants, or one bacteria and the other bacteria, is having the same antigen. That is, and these antigens should have the same epitope. Epitope is a receptor over the antigen. You remember, right? What I told epitope is. Yeah.
Binding site.
Pardon?
It's the antibody binding site in the antigen.
Yeah, it's on the antigen. Yeah. Good. Okay. So in this heterophyll antigen, these are the antigens of different species, but they share the same epitope. An antibody produced against antigen of one species can react with other and vice versa. That is, when these antigens enter the human body, the antibody of produced against these antigens can react with the antibodies of the antigen of other species also. So these type of antigens are called heterophyll antigens. You'll get to know, you'll understand when doing the example time. You just see Weil-Felix test. Weil-Felix test is for typhus fever. Typhus fever is rickettsia. The antigen is rickettsia antigen. So the antibodies produced against rickettsial antigen is used antigen using protease antigen. See, protease antigen is injected. In this test, no protease antigens are used, and this protease antigen is used for detecting antibodies against rickettsial antigen. It's like in Waves test, no, the test tube will be having protease antigen, and we are using this protease antigen to detect the rickettsial antigen.
So it is
So,
like using one antigen to detect the other.
yeah, that's the thing. So protease antigen we are using for detecting the antibodies against rickettsial antigen. So typhus fever is caused by rickettsial agent. So we can't detect the rickettsial agent. So what we are using? We are using protease antigen in the test tube, and we are taking the serum of a human getting infected with typhus fever, and we are trying to find the antibody against rickettsial disease using a protease antigen. Got it now? Yeah. So it is like the antibodies against antigen of one species that is antibody against rickettsial antigen can react with the antigen of a different species. That is protease antigen. And it can occur vice versa also. So antigen of rickettsial antigen can react with the antibody of the protease antigen. Got it now? Yeah.
Somewhat.
Yeah, when you read it again and once or twice, you'll get to know about it. I'll give you one more example also. False bundle test. False bundle test is mainly used for infectious mononucleosis, that is caused by Epstein-Barr virus. So antibody against Epstein-Barr virus is being detected using a sheep RBC antigen. So sheep RBC antigen is used for detecting antibodies against Epstein-Barr virus. Got it?
Okay.
Yeah. So next is call agglutination test.
Basically, this other antigen is used to find the antibody, right?
Yeah, find the antibody, because we need to detect actually these two tests which we are doing right now, like Paul Bennett's test and Waves test. We are using antigen to find the antibody
All right.
mainly. So cold agglutination test and Streptococcus or Mycoplasma MG test is used. These tests are used for primary atypical pneumonia. For cold agglutination test, human O blood group RBCs are used as an antibody detector. Human O blood group RBCs are used, and for MG test, MG is actually Miller group. For MG test, Streptococcus MG antigen is used. Yeah, tell me.
No, sir.
Yeah. So mainly, what you understood is the same thing. In order to find the antibody of a particular disease, mainly we use an antigen of a different species. That's the example of these tests. Got it till now?
Can you repeat that sentence one more time?
Yeah. See, mainly to find the antibody against a particular disease, we use antigen of a different species
Okay.
in the test. So in cold agglutination test, we use antibody in order to find the antibody against primary atypical pneumonia. We use human O blood group RBC antigen. And in order to find antibody against primary atypical pneumonia, we use antigen of streptococcal MG. MG is actually Miller group. Okay, so you got till now, right? Yeah. Okay. Next is adjuvant. So what is an adjuvant? It's any substance that enhances the immunogenicity of an antigen. Usually, it is added to vaccine to increase the immunogenicity of a vaccine. Example is DPT vaccine, right? In
Yes.
DPT vaccine, diphtheria and tetanus is the only main vaccine. Pertussis is actually added to increase the immunogenicity of the particular DT or a tetanus vaccine. So when we are giving the vaccination, pertussis also will work. But when these three are combined together, it becomes much more effective vaccine. So in this, pertussis is acting as an adjuvant. And you just see for influenza vaccination, if your influenza vaccine is combined with a diphtheria toxin and a tetanus toxin, it becomes more potent vaccine. So here DT and tetanus vaccine, toxides are the adjuvant for influenza vaccine. The next other adjuvants are alum, mineral oil, then the prunes adjuvant, that is the complete and incomplete prunes adjuvant, then Mycobacterium bovis, then silica, beryllium, then thiamarcel is also used as an adjuvant. In multiple choice, they might ask you like, which of the following is not an adjuvant? So just this is the simple adjuvants, just remember this adjuvants. So next is biological class of antigen. So in biological class of antigens, depending on the mechanism of inducing antibody, antigen is classified into T cell dependent, T cell independent, and super antigens. So the class of antigens are divided into three types depending on the mechanism of inducing the antibody. So the three types are T cell dependent, T cell independent, and super antigens. You know why they are called T cell dependent, T cell independent, you'll come to know in the further. So in T cell dependent antigen class, so first the antigen enters in the body. Once after the antigen enters the body, is taken up by the antigen presenting cells. Antigen presenting cells, they present the antigen to the T cell. Why they are called antigen presenting cells? Because they present the antigen to the T cells. So they are called antigen presenting cells. And once the antigen is presented to T cell, the T cell gets activated and they secrete cytokines. And these cytokines, they go and activate the B cells, and the B cells form plasma cells, and the plasma cells produce antibody. So first antigen enters the body. Once antigen enters the body, is taken up by the antigen presenting cells, and these antigen presenting cells present the antigen to the T cells, and T cells gets activated and they secrete cytokines, and these cytokines stimulates the B cells and they form get activated into plasma cells, and then the plasma cells produce the antibody. So you got why they are T cell dependent, because their T cell is present in the pathway, and in order to produce antibodies, they request the T cell activation to secrete the cytokine. So now you understood why it's called T cell dependent, right? Yeah. Okay. Now the other one is called, one more class is called T cell independent antigen. So why is called T independent? Because they bypass the antigen presenting cells and T cells. That is, these two antigen presenting cells and T cell activation is not there. These antigens directly binds to the B cells and stimulates it polyclonally. This binding is a non-specific binding, and hence increased secretion of non-specific antibodies, that is hypergamma-globulinemia is seen. These antibodies produced is not specific to a particular antigen. Whichever antigen comes and whichever the body finds as an antigenic or a foreign substance, it attacks everything. So it's non-specific antibody, and the binding is non-specific binding because it binds to any antigen or a foreign substance. So now it is T cell independent because it surpasses the stage of antigen presenting cell and T cell. It directly binds to B cell, and the B cells polyclonally forms is stimulated and it produces non-specific antibodies. Next we come to the super antigens. Super antigens is the one where the antigen directly binds to variable beta region of the T cell receptors. It is not processed here. No, it is not processed by the antigen presenting cells. It directly binds to the variable beta region of the T cell receptors. The binding is non-specific here also. It's not non-specific. It's not specific. It's non-specific. It causes massive release of cytokines called as cytokine storm. They might ask you what is cytokine storm. It is found in which type of antigens? That is super antigens. Cytokine storm is seen in super antigens, which as it causes massive release of cytokines. So these cytokines causes an inflammation, and these cytokines includes inflammatory mediators, and inflammatory mediators are interferons gamma, interleukins 1, interleukin 6, tumor necrosis factor alpha, and tumor necrosis factor beta. These are the main inflammatory mediators, and these inflammatory mediators causes activation of B cells. That's the polyclonal. Same, I told you polyclonal activation of B cells, and there is increased secretion of non-specific antibodies as hypergamma-globulinemia, and it is non-specific polyclonal antibodies. So you got it right? Why it's called super antigens?
Yes.
Yeah. It's called super antigens because there are massive release of cytokines, and it causes cytokine storm. So this is the like image of the different which I told you, like T cell dependent, and T cell independent, and super antigen. So above one is a T cell dependent antigen. It is taken up by the antigen presenting cells, and antigen presenting cells express it to the T cells, and T cells then release cytokines which activates the B cells. Blue color is the B cells, and these B cells causes release of specific antibody production. Whereas super antigens, they don't stimulate, are not present to the antigen presenting cells. They directly to the variable beta region of the T cells, and these T cells causes massive non-specific cytokine release, and this non-specific cytokine release activates the polyclonal B cell activation, and it causes release of non-specific polyclonal antibodies, that is hypergamma globulin Nu. And down one is the T cell independent antigen. There is no activation of antigen presenting cells, neither a T cells. It directly binds to the B cells, and it's a non-specific binding. It polyclonally activates the B cells, and it causes release of hypergamma globulin Nu. So this image you keep in your mind, then it will be very easy for you to classify the antigens. So examples of they will ask you what is our examples of super antigens. Super antigens, examples of bacterial super antigens are staphylococcal toxin. Staphylococcal toxin, main importance are toxic shock syndrome toxin, exfoliative toxin, and enterotoxin. Again, when you treat staphylococcus, we will again come into these topics. Then next is mainly staphylococcus is the one bacterial super antigen. Then viral super antigen is Epstein-Barr virus associated super antigen, cytomegalovirus virus super antigen, rabies nucleocapsid, then HIV encoded super antigen. Then in fungal, it is the furfur antigen, Malassezia furfur. So they might ask you, give an example of few super antigens. These are very important. So next is this: what are the disease associated with super antigen? Example is toxic shock syndrome. Then next is food poison is due to the enterotoxin mainly. Then scald skin syndrome is exfoliated toxin of staphylococcus. Then atopic dermatitis, Kawasaki syndrome, psoriasis, etc. are example of super antigen diseases caused by the super antigens. So now antigen topic is over. Now we'll come to the antibody. So antibody or immunoglobulin is a specialized glycoprotein. They might ask you what is the content of a antibody? It's a specialized glycoprotein. It's produced by activated B cells, that is plasma cells, in response to an antigen. So you got it? So that I told you, like the B cells produce they convert into plasma cells, and it's a plasma cells which produce the antibody. So in response to an antigen, and this is capable of combining with the antigen that triggers its production. There are 5 classes of immunoglobulins, that is immunoglobulin G, A, M, D, and E. And these classes of immunoglobulins is classified. It's called IgG, IgM, Ig, D, and E, and it's based on the Greek letters. That is the Ig, alpha, beta, alpha, mu, gamma, then epsilon, and alpha, mu, gamma, delta, and epsilon. Yeah. So now we'll come to the structure of an antibody. So this structure of an antibody is commonly seen. You have seen it multiple times. You remember, right? There is Y-shaped structure of an antibody. Y-shape is a heterodimer. It's composed of 4 polypeptide chain. It has got 2 light chain and 2 heavy chain. 2 light chain are on the outside and 2 heavy chain on the inside, which is the full length. The smaller length one is called light chain, and the longer length is one heavy chain. This outside or inside it can be any from any side. Don't think like light chains are always outside. It can be inside also. There are 2 light chains. The molecular weight of light chain is 25,000 Dalton each, and heavy chains are 50,000 Dalton each. So now we come to, there are 2 light chains. These light chains can either be kappa or lambda. There are 2 types. There's kappa or lambda. It is either both are kappa or either both are lambda, but it cannot be either kappa lambda. So these 2 light chains will be either both of them are kappa or either both of them are lambda. The next is heavy chain. There are this I told like this IgM, IgG, IgA, IgD, and IgE are classified based on the heavy chains. So there are 5 classes based on amino acid sequence of the constant region of heavy chain. It is called as an isotypic variation. This is the variation type of heavy chain in each immunoglobulin class and the heavy chain type. So based I told like gamma is IgG, alpha is IgA, mu is IgM, delta is IgD, and epsilon is IgE. So you got it right. So immunoglobulins are mainly classified not on light chain. It's mainly classified due to the constant region of the heavy chain, based on constant region of heavy chain. Got it?
Okay.
Yeah.
Yes.
So they might ask you, like immunoglobulin is classified based on light chain or a heavy chain. Always remember, is a heavy chain. So variable and the constant. See, this light chain and heavy chain has 2 regions. There's a variable and the constant region. So variable region is the one which is amino terminal end, and it has both light chain and heavy chain as variable light chain and variable heavy chain. So amino terminal end, you write the above part. So you see in the antibody, this amino terminal end. This is the light chain. This amino terminal NH3 is there, right?
Yeah.
Yeah. This one. This is the variable region. So that is the light chain and heavy chain. This is the variable region. So this is VL, and this is VH region. So you got it about the variable region of light chain and variable region of heavy chain. And this amino terminal end is where the antigen binding site. So this antigen binding site FAB is called FAB because the antibody binds to this amino terminal end. So it's called FAB.